All right, let's get started. Welcome everyone to the first of two ISHR JMCC cardiovascular webinars for this week. Today's webinar focuses on early career investigators. We actually have three award-winning early career scientists who have recently received ISHR awards in various categories who will present their research for you today. I'd like to first again thank the JMCC for sponsoring today's webinar. And point out that we are currently seeking submissions on topics of special interest, which you can see here. We have two topics of interest right now: metabolism and inflammation, and cardiac fibrosis, regeneration and repair. So please check out the JMCC website for more information. And before we get started, I will just ask everyone to enter any questions into the Q&A box. I will have one Q&A session at the end of all three talks. And so with that, it's my great pleasure to introduce today's co-moderators for our session, Dr. Litsa Kranias of the University of Cincinnati and Dr. Chen Gao of UCLA. So I'll turn the floor over to them. Thank you again for attending and looking forward to a good discussion.
Thank you very much, Ron, and good morning to everybody. I am Litsa Kranias, or good afternoon. I am Litsa Kranias, and Chen, would you like to say a couple of words?
Hi everyone. Thank you, Ron, for the nice introduction. I'm Chen Gao from UCLA. I'll be Lisa's co-moderator today.
Yes, and we would like to start today's session with Adrian, Dr. Adrian Arrieta, who received the 2020 ISHR North American Award, the Litsa Kranias Early Career Investigator Leadership Award. And if I can take a couple of minutes to talk about the purpose of this award, and this was written by our early career scientists. We had nothing to do with it, but the award recognizes the exemplary effort and initiative of an early career investigator in promoting the mission and the goals of the ISHR, specifically in the areas of mentorship, collaborative scientific discovery, and service to the ISHR North American Society community. So this was my great pleasure, and to congratulate Adrian for receiving the award. So Dr. Arrieta did his PhD degree in the laboratory of Chris Klambowski at San Diego State University, and his research focused on ER protein folding and misfolding, especially in response to stress in cardiac myocytes and following ischemia reperfusion injury. Currently, Dr. Arrieta is a postdoctoral fellow in the laboratory of Dr. Tom Wodriska at UCLA, and he's working on mechanisms of protein quality control, and also in the context of chromatin remodeling and the associated dynamics in heart disease. Dr. Arrieta also received the 2021 JBC Herbert Cabot Early Career Investigator Award for his doctoral research, and we congratulate him for that. And he's currently funded on a T32 award between UCLA and Caltech on integrative cardiometabolic medicine. Adrian, we thank you very much for being here today, and we're looking forward to your talk.
Thank you, Dr. Kranias, and thank you, Ron and Chen, for moderating the session, and thank you to the JMCC and ISHR NAS section for allowing me to present my work today. So I'll be talking about that specific research that I performed during my doctoral studies in Dr. Klambowski's laboratory. So that work was this one. That's not it. Sorry, it was this one. So that story is about a particular protein termed mesencephalic astrocyte-derived neurotrophic factor. And in Dr. Gombaski's laboratory, I studied how this ER-resident protein protects against reductive stress in the heart, which is a very, still a very nascent concept, but I'll get more into the specifics behind that. So as a little bit of background, as Dr. Kranias had mentioned, we in Dr. Gombaski's laboratory, we have focused on the endoplasmic reticulum as a critical site of protein synthesis, folding, and trafficking in cardiac myocytes, and the proteins that are processed in this specific compartment are critical to myocyte viability and function. So you think about your favorite calcium handling protein, your favorite GPCR, your favorite myocyte signaling protein has a very high likelihood to have been made in this compartment. And so this compartment is also filled with the protein folding machinery that helps these proteins achieve their functional conformations. And when that balance of protein folding chaperones and their clients is disrupted, a condition known as ER stress occurs. And so this is when you have an accumulation of misfolded proteins, and that can cause the rest of the machinery within the ER to become disrupted in its function. And so this condition of ER stress can be brought about by changes in a few different variables. This is not an exhaustive list, but that can include pH, redox potential, changes in ER calcium. This is where much of the cellular calcium is stored. You can also have a disruption in the concentrations of nutrients and oxygen in this compartment. And if this is not resolved, then this can ultimately lead to myocyte death. And so the prototypical example, as it were, of an ER stress condition in cardiac myocytes is ischemia and reperfusion injury. And so this is where you have a blockage in one or more of the major coronary arteries in the heart, and this can prevent blood flow to that specific part of the myocardium, leading to ER stress in that part of the tissue in those myocytes. And as a result, these myocytes not survive. And this ER stress can also be worsened by the onset of reperfusion. So this is where someone will go into the cath lab, have those clots removed, and you can have a restoration of blood flow. But this can cause rapid variations in concentrations of nutrients and oxygen. As we know, that's one of the major damage-inducing factors during reperfusion injury. And so the cardiac myocytes in the heart are not without an ability to withstand ER stress, which is to say they can adjust their conditions to try to combat and survive. So this ability is due in part to the endoplasmic reticulum stress response. And this is predominantly, although not completely, mediated by activating transcription factor 6. So ATF6, through mechanisms that are unknown, as an ER transmembrane protein can sense misfolded proteins accumulated in the ER, and upon sensing them will traffic to the Golgi, where it's clipped by some resident proteases to liberate this 50 kilodalton fragment, which mobilizes the nucleus and induces a gene program that helps restore ER protein folding homeostasis or proteostasis. And this is the reduction of chaperones, protein degradation machinery, and protein disulfide isomerases, which help form disulfide bonds in some of the nascent proteins that traffic through the ER. And so when Dr. Gombaski was beginning to study ATF6 and cardiac myocytes, one of the first things amongst many that he did was generate this transgenic mouse, which contains the 50 kilodalton fragment of ATF6 fused to the mutated estrogen receptor, driven by the alpha MHC promoter. And so this is tamoxifen inducible. And so he used this mouse to ask the question, what genes are transcribed, induced by ATF6 in cardiac myocytes? And so there were two groups of ATF6 regulated genes that came to be elucidated by this approach. And so there was as it were, these classical ER stress inducible genes, kind of as it were, kind of like the positive controls of ER chaperones and ER associated degradation proteins. And so the prototypical example being GRP78, which Dr. Gombaski's laboratory had previously shown is induced during simulated ischemia. But the more surprising thing was that in addition to these targets, ATF6 also upregulates genes that don't really have anything to do with ER stress per se, but are still protective during cardiac injury. And so the poster child of this observation was a gene encoding the antioxidant protein catalase, which is one of the strongest antioxidant proteins known to man. given its high turnover rate of hydrogen peroxide, which is generated during reperfusion injury. And so this led us to consider that, okay, well, ATF6 can be active during both of these aspects of the injury. And so this, amongst these genes that were regulated by ATF6, one of the ones that caught our attention was mesencephalic astrocyte-derived neurotrophic factor, which from now on onwards are referred to as MANF. And so this caught our attention for multiple reasons. First, it had not, prior to my work in this laboratory, hadn't really been examined as an ER stress-inducible protein, definitely not studied in the heart. But importantly, it had no structural homologs and an unknown function, prior to this study. But a couple of features of this protein led us to think that it has a function specific to the ER. And the first one being that it has this ER signal sequence that definitely allows it to be trafficked to the ER and processes an ER resident protein. But importantly, it has this C-terminal RTDL, which Dr. Nicolás' laboratory would go on to show acts as an ER retention signal. So this tells us this is a protein that very likely exerts its role within the endoplasmic reticulum. And so to first figure out whether this protein is important in the context of ischemic reperfusion injury, one of the first things we did was generate, oh, I'm sorry, I can't see some of my stuff because of Zoom. So this led us to the overall hypothesis, that during ischemic reperfusion injury, which results in ER stress, ER localized ATF6, which then becomes active, can upregulate MANF, and this leads to cardiomyocyte death, inhibits cardiomyocyte death via the function of MANF. And so that was our big question: is what exactly is MANF doing in the ER that could be important, given that also it's a protein of unknown function and of unique structure. So one of the first things in the study was to generate a transgenic MANF loss of function mouse, and to perform this approach, this mouse has a synthetic microRNA targeting the MANF transcript, and this is driven by the alpha MHC promoters, so we can specifically knock down MANF in cardiomyocytes in the heart. And so this represents about an 80 to 90% knockdown in the heart of these mice. And when we digested these hearts and cultured the adult myocytes and subjected them to simulated ischemic reperfusion, we observed a significant decrease in myocyte viability following simulated ischemic reperfusion injury. So this tells us that it's really affecting the cardiac myocytes specifically in this injury model. And when these mice were subjected to ex vivo ischemic reperfusion injury, we observed a significant increase in the infarct size in these mice. And we also performed LDH release from the perfusate, as well as contractile assays to further confirm this phenotype observed here, although I'll get a little bit more into that. But importantly, what we were able to do was take these mice and recapitulate or restore MANF expression via sorry, get a little bit ahead of myself. First thing was that this tells us that is really due to ER stress in cardiac myocytes that MANF loss of function is required for myocyte viability. And so in order to determine whether restoration of MANF expression is sufficient to reverse this phenotype, these mice were injected with an adeno-associated virus expressing flag tag MANF, and we observed that we were able to successfully re-express this flag tag form of MANF and restore expression of this protein to approximately equal expression levels compared to the wild type. And when we take these mice and subject them to the same ex vivo IR injury, we observe a restoration in the percent infarct size, confirming for us that, okay, this protein is really protective in a MANF loss of function model when it's re-expressed. And this was further supported by, as I mentioned, the left ventricular developed pressure contractile function assay, where we observed the restoration and cardiac contractility. So here we have the MANF knockdown with the AV control. And when we re-express it, these mice act pretty similar to wild type, telling us that MANF is necessary and sufficient to protect against ischemic reperfusion injury. And we think this would be through ER stress. caused by the injury model. And sorry, I'm just working through Zoom. And so with this phenotypes in mind, we asked the question of, well, what part of simulated ischemic reperfusion injury is Manf exerting its cardioprotective function? And so to dissect this further, we turned to a model of culture cardiac myocytes, so NRVMs, and we subjected them to simulated ischemia or simulated ischemia followed by reperfusion to figure out during which part of the injury is Manf induced. And so the answer to that is it's induced during both parts of the injury. So that didn't really tell us, okay, well, is Manf protective during one or the other? And so to dissect this further, we knocked down Manf in cardiac myocytes and then subjected them either to ischemia or ischemic reperfusion. And when we look at purely just an ischemic injury model in NRVMs, in this case, cell death was measured by counting the number of PI positive cells, we observed that there's really no difference in the amount of PI positive cells under this injury. However, when we tack on reperfusion, we see a significant increase in the number of PI positive cells in this model system, telling us that Manf is really protective during the reperfusion aspect of this injury. And so to validate that even further, we measured the release of agent GB1, which normally is localized to the nucleus and is released during cellular necrosis, which typically happens during perfusion injury. And this was significantly increased upon Manf knockdown, telling us that more so that this is definitely a reperfusion injury-mediated phenotype. And further supported by the increase in cellular ROS that's generated upon reperfusion. This led us to think, okay, well, is there any indicator that the ER is actually stressed during this reperfusion injury? And we observed that that was indeed the case. Here we were able to observe induction of 3 other ATF6 inducible proteins, GRP94, GRP78, and PDIA6, and these were significantly induced as well. And so put together, this tells us that Manf is specifically protective during reperfusion injury. And so we chose to take that approach to figure out, okay, what is Manf exactly doing? And so to kind of hopefully dissect this more, we turned to the causative agent of reperfusion injury, and we asked whether this agent alone, hydrogen peroxide or ROS, can decrease myocyte viability upon treatment with this agent. And so here we observe that bars are getting in the way. With Manf knockdown alone, we observe that there is a basal decrease in myocyte viability, and that hydrogen peroxide also has its own killing effect in this model. And we put the two together, we see that this effect is approximately additive, telling us that these two injuries, as it were, both contribute to decreased myocyte viability, but did not act synergistically, telling us that it's really not ROS that was killing cells upon Manf knockdown. And so here we go. So this led us to think back a little bit more about how Manf can protect against protein misfolding that's caused during reperfusion injury. And so this led us to really start to dissect this concept of reductive stress. What do I mean by that? So as I mentioned in the beginning, proteins that are in the ER can have disulfide bonds that are required for their functional conformations. And this process of forming disulfide bonds is an oxidative process. And so when you have either decreased oxygen, or you can tack on reducing agents in cell culture to prevent disulfide bonds from forming or reverse their formation. And this becomes problematic in the presence or restoration of molecular oxygen, because that can cause aberrant disulfide bond formations and therefore contribute to ER protein misfolding. And so this led us to think, well, what if this concept of injury due to reductive stress could be that's what Manf is protecting against. And so when we treated anti-Manf transfected cells with DCT as a reducing agent, we observed that this effect was highly synergistic with Manf knockdown, telling us that it's reductive stress that Manf is protecting against. And so to kind of prove that to us that this was the case, we also treated cells with dapsogardin, which is a different form of ER stress. In this case, we compromise calcium handling by inhibiting SERCA. And we see that again, that this effect is approximately additive. And so given that Manf loss of function causes an increase in ATF6 activity through induction via via induction of GRP94, 78, and PDI-A6. We asked whether Manf is able to form complexes with misfolded proteins in the under the context of reductive stress, i.e. DTT. And so to interrogate that, we infected NREMs with our flag type form of Manf along with a terminally misfolded protein, mono misfolded protein alpha-1 antitrypsin, delta CT. And so here, as a proof of concept, we were able to express both the flag Manf as well as do an immunoprecipitation for it. And we were also able to immunoblot for the alpha-1 antitrypsin. But when we do an IP for flag, a blot for alpha-1 antitrypsin, we see that it's only under reductive stress conditions, DTT, that Manf is forming a complex with misfolded proteins, telling us that this is where we might focus our efforts and figure out what exactly is Manf doing during reductive stress. And so this led us to ask, well, if Manf is forming a complex with these misfolded proteins, is it acting in a functional manner? And so one of the best ways for Manf to do that would be to have the same function as a lot of other ATF600 proteins, and that's by acting as a chaperone. And so we turned to two classical assays to figure out whether Manf has a chaperone function, and that's through these aggregation assays. In this case, where we can purify recombinant Manf and probe its chaperone activity by incubating it with alpha-lactalbumin or insulin, which have disulfide bonds, break those disulfide bonds with DTT, and then cause these proteins to aggregate. And then the other assay is to incubate recombinant Manf with heat denatured, unfolded, or inactive citrate synthase. And here we observed that Manf was able to inhibit the aggregation of both lactalbumin and insulin in a concentration-dependent manner. And it's also able to restore the folding and therefore function of unfolded citrate synthase, and it has a similar potency as the other established chaperone that I mentioned, GRP78. So this tells us that Manf is indeed, via classical measures, a novel type of chaperone. And so we wanted to figure out what exactly is giving Manf these chaperone properties, and so we did an alignment to figure out is there any hyper-conserved domains or amino acids within the Manf amino acid sequence. And so it's highly conserved across species, but what really caught our attention, given our observations in the context of reductive stress, we have we're able to observe 8 highly conserved cysteine residues that we think could have a hand in either directly mediating Manf's function as a chaperone, or at least contribute to its structure. And so to interrogate whether these cysteine residues are important, we generated a flag type form of Manf that was either the wild type or with all 8 cysteines mutated to alanines. And we were able to assess whether these cysteines mediate direct interactions with the model misfolded protein alpha-1 antitrypsin. And so we turned back to our flag IP and A1AT immunoprecipitation approach. And so in this case, as I had mentioned previously, we did this under DTT conditions where we know Manf is interacting with misfolded proteins, and we're able to express both the flag wild type and the flag mutant forms along with HA tagged alpha-1 antitrypsin. We're able to.
Excuse me, Adrian, I think we're running short of time.
Okay.
15 minutes per speaker, if we can round that. Thank you.
So I'm actually just about done. So what we were able to observe, I'll jump to this last part, and this is where we're able to incubate recombinant Manf with either wild type or the mutant with model client proteins, and we're able to observe that the mutant form is not able to prevent aggregation of these misfolded proteins, whereas the wild type is able to. And so this tells us that Manf cysteine residues are critical for its chaperone function. And so this work was published in JBC recently, almost actually to a year of the date that it was published. And so these are the people that helped make this work a reality. And so this was all done in the laboratory of Dr. Chris Gambatsky. And of course, I'll take any questions.
Thank you very much, Adrian. Very stimulating start today. We are reserving all the questions for the end of the session. So we will move on with our second speaker, introduced by Dr. Gao.
Yes, our next speaker is Dr. Jose Ángel Nicolás Ávila, who is the recipient of 2020 ISHR NAS Young Investigator competition at the junior tier. Dr. Ávila is currently a postdoctoral researcher with an immunology and heart physiology background. He is currently working in the laboratory of Dr. Ángel Alonso at National Center of Cardiovascular Research, studying myeloid cells and their functions in tissues. He was the recipient of a training scholarship to attend the Keystone Autophagy Mechanisms and Disease meeting in 2020, as well as multiple training fellowships from the National Center of Cardiovascular Research. His recent first author publication, A Network of Macrophages Supports Mitochondrial Homeostasis in the Heart, was also selected as the cover for film. It's a really, really beautiful paper. And with that, I would like to welcome Dr. Ávila.
Sorry, because I had the microphone muted. So thank you, Dr. Gao. I think that you can see my presentation now. Yeah.
Yes.
Okay. So yeah, thank you very much for giving me the opportunity to present our work here today. And in this case, I will present the work related to this project about immunometabolic crosstalk in the heart. So what is this immunometabolic crosstalk? So this is a paradigm that we propose now in which some parenchymal cells, such as cardiomyocytes, rely on immune cells, such as macrophages, to regulate their metabolic homeostasis. And this is based on previous observations from our lab, where macrophages, we see that macrophages are important to maintain the mitochondrial homeostasis of these cardiomyocytes. But not only that, other groups, independent groups, have shown similar things happening in other tissues, such as white adipose tissue, where macrophages are also important to maintain the homeostasis of a totally different cell, such as adipocytes. So in today's talk, I will show a little bit of or the most important experiments of our last work published in Cell, as Dr. Gao just said, but also in the last minutes, I will show some unpublished data about a follow-up study that we are doing right now in the context of cardiac aging. So just as a brief introduction, macrophages are immune cells, but those can be present in almost every tissue at a steady state. And the most important thing about these cells, and the most interesting thing, is that they are important to maintain functions which are critical to maintain the homeostasis of the tissue in which they reside. That's the case of alveolar macrophages, for example, that participate by doing surfactant clearance at the lungs. Or in the case of the intestine, you have intestinal macrophages that mediate the tolerance to food and antigens coming from microbiota. The heart is not an exception. It was published in 2012 that there is an abundant population of macrophages residing in the healthy myocardium. And up to these days, it has been proposed, there have been proposed several functions for those cells, including the modulation of electrical conduction in the auriculoventricular node, but also the supporting function for cardiomyocytes that I will talk to you about in the next slides. So when we were studying those cells, the first thing that we realized is that they are quite abundant in the myocardium and they form kind of a net in the tissue. These cells establish contacts with cardiomyocytes, and to study how much contacts and which kind of contacts these two cell types were having, we developed a model that you have here, where just stochastically we can induce tomato-fluorescent expression only in some cardiomyocytes. So by doing this, we can study the number of interactions, as you can see here, with the cardiomyocyte in red, and we quantify this kind of interactions with macrophages in green, finding that in average, there are between 5 and 6 macrophages establishing contacts with a single cardiomyocyte at once. This was already anticipating important housekeeping functions for those cells, but one of the most striking things that we found in this context were that cardiac macrophages were uptaking material coming from cardiomyocytes. So here you have cardiomyocytes in red, and you see how these cells release small particles that we name cardiac exophores that were about 3 to 4 microns of diameter in average. So this is much larger than an exosome. And just for you to look at this closer, here you have a video from a reconstruction where cardiomyocytes, again, can be looked in red. You have the macrophages in green and the nuclei in blue. And you can see here how this macrophage, which is in contact with a cardiomyocyte, have already incorporated one of these particles. So we started to become curious about the presence of these cardiac exophores in the tissue and which could be the content present in them. So we performed a strategy to isolate those particles and perform proteomic studies. So the first thing that we learned from that was that the content of cardiac exophores was quite varied, including proteins coming from different cellular compartments, such as endoplasmic reticulum, sarcomere, etc. But the vast majority of the protein that we could find there were belonging to mitochondria. And we could confirm that mitochondria were one of the main cargo present in those particles by transmission electron microscopy imaging, but also by immunofluorescence and by flow cytometry, just by using molecular proofs which are specific to mitochondria, such as mitotracker or mitoneer. Also, we found that these particles were accumulating LC3, which is one of the mediators of the autophagy pathway. Specifically, this LC3 is involved in the recruitment of cargo into the autophagosome. So this was already pointing that the formation of these cardiac exophores could be related to the autophagy pathway. And to confirm that, we developed this model in which we eliminate one of the ATG7 alleles specifically from cardiomyocytes. And in this context, what we found was a reduction in the number of cardiac exophores present in the tissue, correlating with less transference of material from cardiomyocytes to macrophages, as you can see here. So then we decided to manipulate this system just by eliminating macrophages. To do that, we use the CD169DTR model, in which upon the injection of diphtheria toxin, we can reduce the number of macrophages up to 80%. So, sorry, up to 20%. This is a 80% reduction. So in this case, we could maintain the depletion for up to 3 weeks. And at this point, what we found was that the cardiomyocytes look very ugly. They started to accumulate aberrant mitochondria with reduced crystal density, as it is quantified here, and you can see here. But not only that, we confirmed that those mitochondria were defective at ATP production by doing ex vivo assays for ATP production in the presence of different substrates. And at the end, what we found was that those mice started to uptake more glucose, as measured by FDG uptake. And finally, these mouse show a progressive reduce in diastolic and systolic function. However, as I told you at the beginning, macrophages do much more things than only phagocytose. So we wanted to have a better model to study this. So we decided to move to the MERTK knockout mice. MERTK is a membrane receptor involved in the recognition of apoptotic cells by macrophages. This MERTK, what it does is to bind phosphatidylserine in an indirect manner. And we knew already that cardiac exophores were exposing phosphatidylserine in their membrane. So in the context of this model, we again could observe an accumulation of unfit mitochondria morphologically with reduced crystal density, but also that produce less ATP. And also in the case of these mice, we could observe a switch to a more glycolytic metabolism and the presence of diastolic dysfunction, among other cardiac problems. And finally, other features that were shared by these two models, both the cardiac macrophage depleted and the MERTK knockout mice, was the accumulation of debris in the extracellular space, including mitochondria and mitochondria-derived compounds, as quantified here. And these are very well known to be strong activators of the NLRP3 inflammasome that was increased and activated in both cases. Not only that, we also found that this correlates in time with a decrease in the autophagy pathway. And this has been already proposed that upon activation of the NLRP3 inflammasome, some caspases get activated that cleave components of the autophagy pathway and can even sequester them. So the autophagy flux gets reduced. So these are just some pieces of data, and this is the final model that we propose in our recent paper, where part of the mitochondria that the cardiomyocyte contains upon damage will be degraded intracellularly through canonical autophagy. However, in some cases, part of this pool of mitochondria is released out of the cell in the form of cardiac exophores that are captured and eliminated by resident macrophages in the tissue. When you don't have those cells, or if they are deficient at phagocytosis because they lack some phagocytic receptor, the material will start to accumulate in the extracellular space and could finally activate the inflammasome. This activation of the inflammasome can blunt the production of new particles, new exophores, through the inhibition of autophagy. So now in the next minutes, I will show you just some preliminary data of a follow-up study that we have in the context of aging. And the nexus here is that for those of you that are currently working in the cardiac aging, probably many of the features observed in the cardiac macrophage depleted mice and the MERTK deficient mice will be familiar to you. For example, the accumulation of dysfunctional mitochondria, oxidative stress, inflammation, reduction in autophagy pathway, and diastolic dysfunction are all of them features which are shared with our cardiac macrophage depleted and MERTK deficient mice. And not only that, it's also known that with aging, there is a loss of function of the immune compartment. In the case of the macrophages, it is known that you have a reduction in the number of those cells, but also a reduction of the function of those cells, including phagocytosis. So with these results, we started to consider if cardiac aging could be somehow related with immunosenescence, so a loss of function of the immune system. So the first thing that we did with our cardiomyocyte reporter model was to analyze the phagocytic capacity of the macrophages there. And at 1 year of age, we could not observe any difference in the phagocytic capacity of macrophages in the cardiac tissue. However, what we do find was a strong decrease in the number of those cells per gram of tissue. As you can see here, this is the total number of macrophages, and these are the changes in number for different subsets that have been proposed by other groups. This reduction in the number of macrophages was progressive and correlating time with a reduction in proliferative capacity as measured with staining for Ki67. And the proliferation and the maintenance of macrophage population is known to be controlled by one main receptor, which is the CSF1 receptor. This protein is expressed on the membrane of monocytes and macrophages and has two main ligands. One of them is CSF1 and the other one is IL-34. So the first thing that we did was just to check which cell types were expressing both cytokines in the context of the heart. To do that, we use different public available databases. So Tabula Muricenis in the case of mice and Human Cell Atlas in the case of humans. And what we found was that CSF1 was mostly expressed by fibroblasts in both species in terms of amount of expression, but also the number of cells that were expressing it. And in the case of IL-34, it was quite striking that it seems to be quite restricted to parasites. So with this, we propose a model in which the number of macrophages will be maintained through the production of CSF1 by fibroblasts and the production of IL-34 by parasites. And not only that, these databases contain information about the age of the different specimens, even mice or humans. And we found in both cases that there is a reduction in the levels of expression of both cytokines with age. Finally, we developed a gating strategy to identify those stromal populations by FACS, finding that it's not only the level of expression of these two cytokines, but also the number of the stromal cell populations, such as fibroblasts, pericytes, and smooth muscle cells, change with age. So now we are working with this model, with cardiac aging, and we are thinking of it about like an impairment in this immune metabolic crosstalk which is happening in the heart. In the context of aging, we have less stromal cells and less CSF1 and IL-34 expression that would lead to a decrease in cardiac macrophages that we see in these age mice, and finally lead to a similar phenotype to the observed in the cardiac macrophage depleted mice. Even if these cells at this point does not seem to have an impaired phagocytosis, this is something that we will be checking in different ways in the next months. The reduction in the number of macrophages could be enough to not be able to keep the house clean. So everything, all these changes will lead to a situation in which the material will start to be accumulating in the extracellular space, activating the inflammasome, reducing the autophagy flux, and overall causing this accumulation of damaged mitochondria in the cardiomyocytes. Now we have many tools to abort this from different perspectives, and I will hope to show you more data in the next months. And with this, I would like to finish just by thanking all the people in my lab, especially to my boss, Andres Hidalgo, and Maria Sanchez, Laura Pena, and Sofia Vieto, which are working right now in different follow-up studies from this new concept in the lab. Also, I want to thank to all our collaborators and funding agencies. And thank you for your attention. I will be glad to answer any question that you have.
Beautiful presentation.
Thank you.
Thank you so very much. And we are ready to proceed with our last speaker of this morning, Dr. Shrike Zhang, who is an assistant professor at Harvard Medical School and an associate bioengineer at the Brigham and Women's Hospital. His research interests include 3D bioprinting, organ on a chip, biomaterials, regenerative engineering, bioanalysis, and of course, biology. His scientific contributions have been recognized by over 40 regional, national, and international awards. He has been invited to deliver over 120 lectures worldwide, and he's an envious reviewer of many journals around the country. Dr. Zhang recently received the Materials Today Rising Star Award and also the Chinese Association for Biomaterials Young Investigator Award. Congratulations, and thank you for being with us today, Dr. Zhang.
All right, thank you very much, Dr. Kranias, and thanks for the invitation. So, yeah, today I'm going to talk about some of our work in the past few years on basically high content emulation of cardiovascular tissues in particular. So a little background to start with, we have been really working on different aspects of research, and one of these is basically in vitro emulation of the human tissues or human-based tissue models for drug testing. So rationale for that is a conventional model, as we know, being two-dimensional static cell cultures or animal models don't really recapitulate how the human systems are behaving or responding to drug molecules. So leading to a lot of failures in the development pipeline. So then that's why in the past 10, 15 years, people have been really interested in how we can emulate or build these in vitro human tissue and organ models in a high content manner to allow better screening of drugs in like for their responses in human systems. So these are so-called organ chip systems. So these are little micro devices where you can host the micro tissue models in there, and they are very, very relevant to the in vivo counterparts. And then also they can put them together in the way that these different organs are linked in human systems. So you can now start also to look at their interactions in the dynamic manner. That's something that you can never really do with conventional two-dimensional models. And then using these models, you can start to look at either drug development or person's medicine to combine with human patient-derived cells for improved applications. So then, as you can also imagine, all those techniques that I described in the previous page can be readily adapted to different other areas too. One of them is tissue engineering and genetic medicine, basically how you can engineer really functional human tissues and organs and put them back into human system for regeneration purpose, right? So then this is more longer term goal, because in this case, you have to make sure that everything's coming together in the right way, in a biocompatible manner without any other issues before we put everything into the human system. But eventually this is going to allow us to really solve the organ shortage issue that we face right now in the translational kind of area. So then with those in mind, our lab has been really interesting. Quite a wide variety of technologies, including, for example, biomaterials and to biofabrication, in particular bioprinting, and all the way to biomedical devices and bioanalysis, as mentioned. And one of the examples is we have been pioneering, for example, the combination of bioprinting together with bioreactors to allow better emulation of human tissues and organs in vitro than they were possible before. So a few examples. One is we have been engineering these high content bioreactors that allow dynamic cultures of the cells or microtissues in there to allow a better emulation of the tissues. And you can, of course, as I mentioned, place all these different organoids or microtissues together in a way that they are arranged in human system to look at their interactions. So I'm not going to go into details about this, but just showing the overall capacities that we have been building in the lab in the past years. And then, as I mentioned, the biofabrication is another area. So besides the conventional biofabrication methods that are usually based on manual or semi-manual operations, we have been particularly interested in bioprinting, which is a three-dimensional spatial deposition method that allows patterning of cells and biomaterials in a well-controlled manner. So this is going to give you not only structural flexibility in building the tissues, but also high spatial controls and also larger scale fabrication with automation capacity, further ensuring the batch-to-batch reproducibility of tissue that you make over time. All right, so then within the bioprinting field, we have also been working on actually pretty much every single aspect of bioprinting. So for example, we have been working on multi-material bioprinting, basically how you can put multiple cell types together and multiple ECM together in a way that the different cells and ECMs are basically arranged in vivo to allow better emulation of these three-dimensional constructs of human tissues and organs. And then we have been working a lot of things on tendon tissue bioprinting, basically bioprinting of these hollow microtissue constructs that are widely relevant in the human system, for example, the vasculature and all the other hollow tissues that are present in the human system. So then we have also been working quite intensively on bioinks and bioink processing methods, because again, bioinks, which basically refers to the combination of biomaterials and the cells, they are actually very important in bioprinting, because they not only have to support the or enable bioprinting in the first place, but also at the same time, they have to be really able to allow the cell and tissue to grow and to function after bioprinting process. So that's actually a very important aspect of things. And then last part is something we call minimally invasive bioprinting or delivery. So basically, if you have a wound surface, sometimes you don't want to print or fabricate something and then transplant, but instead, you want to be able to directly print on the surface of the wound to allow for in situ wound dressing. That's usually giving you some advantages of like time, like fast curing process. So then I'm going to give a few examples today, specifically on how we have been able to combine the on-chip platforms and the biofabrication platforms, each of which has their unique advantages and disadvantages in basically generating in vitro functional human tissues and organs, and with a focus on cardiovascular tissues. So I'm going to, again, give these examples without having to go into details because of the time. But one example is, for example, we have in early stage been building these cardiac spheroids that are containing different populations of the relevant cell types in there. And within these spheroids that you make, you can actually not only see how they are expressing their respective biomarkers, but also how they are keeping their viability for a month-long period when you bioprint these spheroids into a bioreactor for dynamic culture, right? So then these spheroids also contracting spontaneously, basically also showing the transduction of the signal from one side to the other side of the spheroid. And more importantly, if you use this device to test drug molecules, we did show that if you apply these different molecules that are initially on the market, but then record later on due to cardiotoxicity, we did show that all these molecules or compounds have actually shown noticeable toxicity in our spheroid-based cardiac tissue model that's actually very translational in essence. So then this is the simplest form you can make. And then a little more complex is you can actually use the biofabrication method called photopatterning to actually pattern these substrates with aligned grooves and ridges. And if you place your cardiomyocytes on top, you can then align the cardiomyocytes in one direction that's very similar to their bundled configuration of the myofibers in the human system, right? So then again, you are seeing quite nice viability of the cells and also their expression of the biomarkers. And of course, I mean, if you look at their contractions, they are also very uniform and showing nice contractions of this cardiac myobundles in these devices. So then going to bioprinting, as I mentioned, this is actually very interesting as well, because with bioprinting, you can then create more complex tissues or structures in there. So one example is that you can use bioprinting to create these embedded vascular structures that are not only perfusible, like basically structure relevant, but also biologically relevant by seeding the endothelial cells within these, like on the surface of these lumens, right? So then that's going to actually allow eventual formation of this endothelium on these channels that can be either straight or branching, depending on what bioprinting method or configuration that you use. And then, yeah, so once you have these endothelial cells on top, they are also forming very nice endothelial monolayer, showing nice junction proteins. And if you actually compare these endothelial cells or endothelialized microchannels with a microchannel that they bioprint but not endothelialized, you can actually see improved permeability profiles of these endothelialized microchannels showing their barrier function. So that's actually very important, indicating the biological behaviors. So then if you culture these endothelial cells for long enough, you can also observe the secondary sprouting of the endothelial cells into the surrounding matrix, forming these multi-scale vasculature that's not typically easy to formulate with conventional biofabrication methods. And then in one of our recent demonstrations, we further showed that you can shrink these vasculature down to single capillary level, which may be useful for some of the applications, like when you're trying to emulate very small vessels in the human system. All right, so then not only can you model healthy vessels, but you can also model, for example, disordered vascular systems, right? For example, in this case, we actually infused the human whole blood into these bioprinted endothelialized microchannels, and then we're able to create these thrombosis on chip device. And if you look at their histology, right, so our model versus real human thrombosis, you can see they're very similar to each other, indicating the like pathological relevancy of the model that we have been making with the bioprinting method. All right, so those were tissue embedded vessels, as you can imagine. So basically you have a vascular structure embedded within a tissue. surrounding the vessel. So can you actually make standalone vessels or hollow tissue that are also widely present in the human system, right? So in this case, we actually use the coaxial bioprinting method to just do that, right? So you can basically print these standalone hollow vessels, and you can very easily tune how many layers of walls you have for the vessel, single layer, double layer, or even more layers. And then by tuning the bioprinting conditions, you can also very conveniently modify how big the vessels are, for example, or how thick the walls are, depending on your application. So then with bioprinting, I mean, the three-dimensional deposition method, you can then start to also build three-dimensional vascular bed within this, using this method. So this is actually bioprinted 10-layered vascular bed, and the entire structure is actually perfusible, just like the vascular bed in your system. All right, so then if you further tune the bioprinting parameters, you can then also build in tortuosity, right? So convolutions of the structures in there, for example, when everything's matched, it's straight channels. But then if you start to mismatch the parameters, then you can eventually build this very tortuous vasculature that's also potentially relevant to modeling some of the tissue configurations. All right, and these tortuous vessels that you print, like 10-layer here too, they're also perfusible, believe it or not. And then coming back to the cardiac system, so what you can do with this vascular bed that you print, right? So one thing you can do is that you can actually seed another cell type into surrounding interstitial space of these fibers, in this case cardiomyocytes. So now you can create very similar myocardium tissue that's endothelialized or vascularized with a in vivo counterpart. For example, by controlling how the fibers are oriented, you can actually start to orient the cardiac cells as well. And then these are basic three-dimensional myocardium tissue that are containing the interspaced vasculature, which is actually quite relevant to the in vivo counterpart, having the three-dimensional myocardium with interspaced vessels. Right. So here's some data showing that basically you can see very nice lining of the endothelial cells within this bioprinted vessel-like structure or vascular bed. And then these are GFP labeled, but then you can also see the spontaneous and synchronized contraction of the cardiomyocytes on the surrounding surface or in the surrounding space. That's on label. We can see their contractions, and if you look at their biomarker expressions, very nice expressions of the proteins. But also, if you look at different regions, you can also synchronize the contraction of these cardiomyocytes, indicating it's an integral piece of the cardiac tissue or a vascularized cardiac tissue that you can make. So then, finally, if you can bring this together with a piece of bioreactor, as I mentioned, right? So this is not only giving you the chance to model the three-dimensionality of the cardiac tissue, but also you can perfuse the entire structure, also giving you dynamics, right? So this is going to not only give you the perfusion capacity, but also it's going to improve the oxidation profiles of these microtissues in 3 dimensions. And this is going to effectively increase the viability of both endothelial cells and cardiomyocytes. And then eventually you can put them as a system for drug screening, for example, looking at how the cells are responding to different drug molecules you put into the system. All right, so with that, I'm going to just end with a couple more slides here. I didn't mention this before, but we have also been working on a range of biosensors that are allowing integration with these microfluidic devices to enable in situ continuous and long-invasive monitoring of this organ microtissue responses and behaviors. So then with everything coming together, you can start to look at how you can link different things together and study either drug screening or improve the tissue maturation with bioreactors. So one example is you can not only generate these healthy cardiac tissues, but also you can generate these cardiac tissues with preconditions, for example, fibrosis. And then with that in mind, you can then study all the, for example, anti-cancer drug efficacy and safety in the context of patient-specific manner, right? So with that, I'm going to end this by thanking all the wonderful collaborators and the funding agencies and, of course, all the wonderful lab members that we have been having in the last few years. Thank you very much.
Thank you very much.
Thank you.
Yeah, go ahead.
Thank you so much, Dr. Zhang. Now we are open for questions. We are running out of time, so maybe 1 or 2 really quick questions. The first one is listed in the Q&A boxes by Daniel, and then this question is to Adrian. Do ventricular cardiac macrophages form gap junctions with cardiomyocytes?
Yeah, I think that this one is for me.
I think that's for you. Yeah.
Yeah.
Yeah, we have not checked it, but there is another paper from Cell about cardiac macrophages where they show that those cells establish gap junctions through connexin 43, at least in the auriculoventricular node. I do not remember by heart if they have explored the ventricular myocardium, but I think that at least by qPCR, they show the expression of connexin 43. This paper is from Hulsmann and the Matthias Narendorf laboratory.
Wonderful. We have another question from Edward Depkov. Considering the large number of residential macrophages in your image, would it be possible to provide the percent of macrophages in relation to other interstitial cells in the myocardium? For instance, endothelial cells, fibroblasts, pericytes, and so on.
Yes, it will be possible, but the major problem that we found there is the availability of markers that are unique for those populations, because many of the markers between fibroblasts, pericytes, smooth muscle cells are shared. So it's very difficult without having mouse models, reporter mouse models for those cell types to do this kind of labeling of the cells and then do the quantification. But we are trying to do that.
Wonderful. Chen, would you like to read the last one?
Yes. So I believe this is also for Jose. Thank you. Do you envision other kinds of interaction between cardiomyocytes and the macrophages besides phagocytosis of vesicles?
One of them is the one that Daniel already asked for, the formation of gap junctions, that in this other Cell paper, it's proposed to modulate the membrane potential of the cardiomyocytes and the conduction of the electrical impulse. But also we are quite interested in the potential growth factors that the macrophages can produce in the cardiac tissue. So we know that cardiac macrophages produce, among other things, the IGF-1, the insulin growth factor 1, which in the context of the brain, it has been proposed to be expressed by the microglia and to be important to modulate the metabolism of neurons. And also we are interested in the PDGF growth factors because some of the isoforms are also expressed by macrophages in the cardiac tissue. And even some of them one of them seems to be quite specific for macrophages, at least by qPCR. So yeah, probably these macrophages communicate with the stromal compartment, but also cardiomyocytes through the release of growth factors and other cytokines.
Thank you. Thank you so much. As we are running out of time, I would like to thank Litsa and all our speakers again for sharing the great science and really showcase the future of our cardiovascular community at ISHR. And as our 2021 ISHR conference will be held in Denver, I would like to encourage everyone, especially the early career investigators, to participate in the conference and apply for the award, the Young Investigator Award and Lisa Kranias Early Career Leadership Award. And we look forward to meeting everyone soon, hopefully in person at Denver.
Again, we thank you very much for this participation. We had amazing talks this morning. You're all the stars, the rising stars in the cardiovascular field. And as Chen said, we feel so proud that our society is in good hands for its future. See you pretty soon in person. Thank you very much to all the attendees as well and the stimulating session that we had. Ron, any closing words?
I'll just say thank you again to all the participants and to our moderators. Definitely check out the ISHR meeting in Denver. I hope to see everyone out here in September. And just really quickly, we have one more webinar on Wednesday on MEP1 alpha and inflammation and fibrosis. So please check this out as well. You can find the registration link here or on the JMCC website. Thank you again for taking part in today's session.
Thank you, everybody, and goodbye.
Bye-bye.
Bye-bye.
I thank you.